We're now going to look at two post-Darwinian 19th century individuals, starting with Ernst Haeckel. Now, those of you that have had biology probably associate him with something called the biogenetic law, and indeed, that is what he is best known for. And if you've not heard of that law by that name, you certainly have heard of it by the way the law is stated, which is ontogeny recapitulates phylogeny. Ontogeny, you'll recall, means development. Phylogeny, of course, means evolution. Recapitulates means repeats. In other words, you repeat the evolutionary stages of your ancestors while you are undergoing prenatal development. So this law, as it were, indicates that, well, we pass through our fish stage because all embryos have gills, and then we pass through our rabbit stage and our you can name anything you want actually until you actually end up being a human being. Unfortunately, well, actually, I think fortunately, that's not the way things work. But a lot of people were buying into this law because of the drawings that Haeckel offered that looked very, very compelling. Unfortunately, they were not based on any firm data. Even Darwin was fooled a little bit by this, not entirely. Here is a good summary by a contemporary individual describing the whole situation with Ernst Haeckel.
Back in the mid-1800s, a German zoologist named Ernst Haeckel tried to prove what we now refer to as recapitulation theory. Basically, and this is not basic at all, recapitulation theory states that ontogeny recapitulates phylogeny. In other words, ontogeny, or the growth and development of an embryo, recapitulates or sums up phylogeny, which is the evolutionary history of a species. So this means, for instance, that a human embryo over the course of its development will go through all of the hundreds of millions of years worth of evolutionary steps that it took for a single-celled organism to evolve into a fully tricked-out person. Haeckel was a contemporary of Darwin, and On the Origin of Species made a giant impression on him, especially a section of it that notes how cool it is that all vertebrate embryos look pretty similar to one another regardless of whether they're mammal or bird or reptile. Darwin, however, cautioned that this probably wasn't a very good way of reconstructing the history of evolution. He just thought it meant that the embryological similarities were evidence of common ancestry between species. Well, Haeckel was kind of a spaz, and he definitely heard the first part of Darwin's idea, but not the rest, so Haeckel jumped onto this idea and very quickly wrote a couple of books about how the development of an embryo mirrors the evolutionary development of adults of a species, which is exactly what Darwin said was not happening. Anyway, Haeckel did spend a lot of time looking at embryos and observed that the slits in the neck of a human embryo resembled the gill slits of fish, which he took to mean that we must have at one point had a fish-like ancestor. He drew tons of figures of different animal embryos in different stages of development to prove his theory, and his illustrations of embryos started to make their way into textbooks all over the world. Haeckel is exactly the sort of person who really ticks other scientists off, because real science-loving scientists like to sit and think about stuff to find out all the problems with an idea before they start publishing books about it. And here Haeckel was firing off volume after volume, and before long all the data he had collected convinced a bunch of other people, including Darwin actually, that he was onto something. But in the end, it turned out that Haeckel was kind of fiddling with his drawings of embryos to make the data fit his recapitulation theory instead of, making the theory to fit the data. But by that time, everybody already knew about the theory, and if there's anything harder than teaching people something, it's unteaching them something.
Which unfortunately is correct, because the biogenetic law is still being taught in many educational institutions, unfortunately including universities, as if it's real, and it's not. I mean, the data that actually show that it's not, some firm data actually go back 1930s, I believe it is, and then on into later on in the 1900s. So the law has been totally discredited. And fortunately, there have been some science articles written about this. I mean, in the journal Science, which is a highly influential journal, showing that there was fraud in terms of how those drawings were made. There's another article, there's no highly conserved embryonic stage in the vertebrates, implications for the current theories of evolution and development. Haeckel was wrong and to some extent a bit of a fraud. Unfortunately, it did influence some other people. I mean, we do have these gills, right? Okay, so let's set the record straight on these gill slits that we have and that fish have. Okay, we did not evolve from fish. That's number one. But what about those gill slits? I mean, it is kind of interesting that we both have them. Well, look what happens to the embryonic gill slits of a shark, for example. So there's the gill slits in the embryo, and they, sure enough, develop into the gill arches of a fish. Fish have gills. But look at the gill arches, look at the what are called the branchial arches of a human embryo. There's some similarity with the fish, but they have nothing to do with gills in us because guess what? We don't have any gills. They actually will develop into all of these other structures, the middle ear bones, the eustachian tube, the carotid artery, the hyoid bone, the larynx, and the neck muscles. Nothing to do with gills.
If you see an early fish embryo and a human embryo side by side, you'll see something remarkable. They look almost identical. We really do look like fish. Both embryos have a head, a body, a tail, and many other similar features.
And one of those similarities exists in the neck, or what's called the pharyngeal area.
In both fish and people, what you find are a series of swellings called gill arches.
Turns out that in fish, those swellings become components of the gill apparatus. In people, they become portions of our lower jaw, portions in our middle ear, part of our voice box.
So, if we're interested in looking at evolutionary lineages, here is a way that makes sense, and it involves the creature that you're seeing here. It's called the ice fish.
It's known as the ice fish. These fish live in the very cold waters of the Southern Ocean, waters that are around 29 degrees Fahrenheit. The most remarkable thing about the ice fish is that they have no red blood cells at all. The only vertebrates, the only animals with backbones on the planet that have no red blood cells. And we know that's because they got rid of red blood cells in the course of evolution. And the signature of that is that we can find, fossilized in their DNA, the remnants of the genes for proteins that make red blood cells red, the hemoglobin genes. So this fish has in its DNA a record of all sorts of changes that have taken place. And as soon as we inspect that, it tells us so much about how these fish are different from their ancestors, and it gives us so many clues about how they've adapted to this very extreme habitat.
So yeah, now that we have DNA analysis at our disposal, more or less, we can start looking at these kinds of things, and in particular, how certain genes are switched on or prevented from being switched on, or in some cases, ones that are switched on and then switched off in the course of development that affect what you end up seeing in a developing organism. And pretty much that's what's going on here with the ice fish. It's just that the red blood cell genes are not switched on in development, and therefore there's no genetic basis for red blood cell development in the ice fish. This is a science that today is called epigenetics, and I already talked about epigenesis. This is a slightly different use of the term because when I talked about epigenesis, I was talking about epigenesis as opposed to preformationism. The science of epigenetics today actually refers to how genes are switched on and off in the course of development that affect what it is you end up seeing in the individual. So it's a slightly different use of the term. It's a science in and of itself. It's huge in molecular and cell biology. And you might also have heard about epigenetics under a different label, EVO-DEVO, EVO-DEVO, evolution development, EVO-DEVO. Pretty much the same as epigenetics. Well, finally, the last person I'm going to mention from this period of time is somebody I mentioned when I was talking about Erasmus Darwin in the other segment, Wilhelm Preyer. He is basically known as the father of behavioral embryology, albeit Erasmus Darwin did have contributions to this area prior to Preyer, so to speak. So, a quote from Preyer, of course translated from German into English: "The fundamental activities of the mind which are manifested only after birth do not originate after birth." Yes, they originate before birth. In other words, there are important events that occur prenatally that affect what happens postnatally. And that's basically what behavioral embryology is all about. So that's about it for this period of time.